Aeromonas veronii biovar sobria. General information. Aeromonas veronii biovar sobria is a bacterium frequently found in aquatic environments and various food products. It is essential to note that while this bacterium can be isolated from stool specimens, its clinical significance requires careful interpretation, considering both laboratory results and clinical presentation. Taxonomy and habitats. Taxonomically, Aeromonas veronii biovar sobria belongs to the Aeromonadaceae family. These bacteria thrive in freshwater habitats worldwide but are less tolerant of saltwater environments. Interestingly, they can also be found in various food sources, such as fresh produce, meat, dairy products, and even in the gut of medical leeches and the mouths of snakes. Clinical significance. Aeromonas veronii biovar sobria can cause gastroenteritis and wound infections in humans, particularly in individuals with compromised immune systems or underlying liver disease. Gastroenteritis caused by this bacterium can manifest as acute watery diarrhea, dysentery, or even a chronic illness. In acute watery diarrhea, stools are loose and typically lack blood or white blood cells. Symptoms and additional considerations. Common symptoms associated with Aeromonas veronii biovar sobria infections include abdominal pain, fever, vomiting, and nausea. Gastroenteritis caused by this bacterium is usually self-limiting, but children may require hospitalization to manage dehydration. While the gastrointestinal tract is a common site of infection, Aeromonas veronii biovar sobria can also be isolated from other sources, most notably blood and wounds. It's particularly important to recognize the potential for this bacterium to cause severe extraintestinal infections, like septicemia and meningitis. Aeromonas veronii biovar sobria. A closer look. Aeromonas veronii biovar sobria is a gram-negative bacterium, showcasing a distinctive straight, rod-like shape with rounded ends when observed under a microscope. This bacterium falls within a size range of 0.3 to 1.0 micrometers in width and 1.0 to 3.5 micrometers in length. In terms of arrangement, Aeromonas veronii biovar sobria cells are typically found as single entities or in pairs. However, in rare instances, they might form short chains. Understanding the gram stain and morphological characteristics of Aeromonas veronii biovar sobria is crucial for its identification and differentiation from other bacterial species. The gram-negative nature of this bacterium indicates its cell wall structure, which influences its susceptibility to various antibiotics. Additionally, its distinct shape and size aid in its recognition during microscopic examination, contributing to accurate diagnosis and appropriate treatment strategies. Culture characteristics of Aeromonas veronii biovar sobria. Aeromonas veronii biovar sobria is characterized as a facultative and aerobic bacterium. This means that it has the ability to grow both in the presence and absence of oxygen. It also exhibits a positive oxidase reaction and is capable of fermenting sugars, a metabolic trait that distinguishes it from some other bacterial species. When cultured on blood agar, Ba, Aeromonas veronii biovar sobria typically forms colonies that are smooth, circular, and convex in shape. These colonies are translucent, meaning that light can pass through them, and they range in size from 1 to 3 millimeters in diameter. A distinctive feature of Aeromonas veronii biovar sobria on blood agar is the presence of beta-hemolysis, which is indicated by a clear zone of complete red blood cell lysis surrounding the colonies. In addition to blood agar, Aeromonas veronii biovar sobria is also capable of growing on MacConkey agar and Brucella blood agar. Growth on MacConkey agar, a selective and differential medium, suggests that this bacterium is capable of fermenting lactose. Growth on Brucella blood agar further supports its ability to thrive in various culture conditions. Please note that the information provided here is based on current knowledge and may be subject to revision as further research is conducted on Aeromonas veronii biovar sobria. Aeromonas veronii biovar sobria, a collection of characteristics, a unique profile, a microbe of interest. Aeromonas veronii biovar sobria is a gram-negative bacterium. Distinctly rod-shaped, these bacilli are medium-sized. thrive in both oxygen-rich, aerobic, and oxygen-depleted, anaerobic environments. Aeromonas veronii biovar sobria's hallmark is beta-hemolysis, a complete breakdown of red blood cells. Tests positive for both catalase and oxidase enzymes. Also, indole positive, but urease negative. Aeromonas veronii biovar sobria exhibits motility, crucial for movement. Shows resistance to the antibiotic vancomycin, however, susceptible to colistin. Notably, lipase positive, indicating the ability to break down fats. Poem. Aeromonas veronii, water's guest, in food, in gut, a subtle test. From leeches to snakes, a diverse day. Diarrhea, wounds, it has its way. Gram-negative rod, beta-hemolysis is gleam. Oxidase positive, a metabolic dream. On blood agar, colonies unfold, a microbe's story to be told. Motile, vancomycin it defies, colistines its bane, lipase its prize. Aeromonas veronii, a name to recall, in the world of microbes, standing tall.